Our next speaker is Adiv Johnson from Tally Health, which is a company that's doing some really exciting stuff, just like all the other talks. Adiv, there you go.
Hello. Is this working?
Yep.
Mic check. Perfect. Aubrey, thank you so much. Really appreciate being here. I'm having a ton of fun. Who's having a fun time? Yeah. Okay, I have a polarizing question for the audience to open things up. It's not that polarizing. How many people here, by show of hand, think we can measure biological age right now? Interesting. Okay. Well, please don't throw too many tomatoes at me, because my talk is going to be about aging biomarkers and what I feel is the unsolved quest to measure biological age. I think this is a really important topic that's highly relevant to RAADfest, because humans are not worms, flies, or mice, you may have noticed. And the reality is we do live a pretty long time. Not as long as we'd like, but we live a long time relative to most animal species, even other primates. And this means that a human lifespan study is not going to happen. It's just going to cost too much money and take too much time. It's not realistic. So if we want to efficiently test longevity interventions at scale in humans, which we definitely do, we need to align on surrogates for biological age that do an amazing job predicting future lifespan and health span. And in this short talk, I'm going to argue that we're making huge progress. This is a problem we can solve, but that it's currently unsolved. Okay, if we want to measure biological age, we should probably first define it. And like many things in the very passionate and opinionated longevity space, the exact definition is going to vary depending on who you talk to. But I think pretty much everyone aligns on the idea that biological age intuitively exists. We all know biological age is real. It explains how an individual is aging internally, externally, and functionally. It explains why people age differently. It explains why you can have remarkable scenarios where someone is 80 or 85 and can thru-hike the entire Appalachian Trail from Georgia to Maine, personal lifelong goal of mine. But other people that are 80 might struggle to walk around the block. These people are biologically not the same age. And importantly, biological age is describing how old someone's body is, irrespective of their chronological age. We're not talking about candles on a birthday cake. We're talking about how old your body actually is. As you might imagine from that definition, this is not a trivial thing to quantify and get everyone to agree that we can quantify. And a major challenge is that aging is a pretty complex, multifarious process that affects molecules, cells, and tissues differently. It's very inconvenient for us scientifically. It would be really nice if it was much simpler. And this is why when you survey longevity scientists, the vast majority of people agree that aging cannot and should not be measured by a single metric because it is multidimensional and heterogeneous. And this is such an important point I want to double click on. We're not going to measure how old our bodies are with one biomarker or metric. There's too much complexity and multidimensionality for that to happen. To give an example of this, I apologize for this terrifying looking person. He was made with Google Gemini. But to illustrate this, let's focus on the most OG biomarker of all time in the aging space, grip strength. Grip strength, of course, reflects musculoskeletal health. It reflects how much upper body strength training someone has done in their lifetime, especially in recent life. It can be measured with a dynamometer or by doing a dead hang exercise, as this terrifying, swole individual is doing. And time again, we see that grip strength is associated with how long people live and how healthy that life is. And this is why it's an aging biomarker, because it's providing more information than chronological age when it comes to predicting age-related outcomes. So there's a signal that's encoded by grip strength that's useful and interesting. But it's very obvious that grip strength doesn't tell the whole story of how you're aging. It's really easy to find someone who has pretty impressive grip strength, but is aging terribly. I started doing a lot of weight training in the past few years. I've met a lot of bros that down energy drinks after not sleeping and drinking, go to the gym. Good for them, but it doesn't mean that they have the healthiest aging and that their biological age is young. So the way to conceptualize grip strength is as a useful biomarker that's shining a very small, incomplete window into biological age. There's so much more information that we're missing. And this is true for every single aging biomarker we have, including VO2 max, the frailty index, circulating blood markers like ApoB, hemoglobin A1C, C-reactive protein, even single question questionnaires like self-rated health and self-perceived aging. If you just ask someone, how healthy are you on a 5-point scale, the answer to that is actually pretty strongly associated with mental health outcomes, physical health outcomes, and how long someone lives. So I would argue self-rated health is an aging biomarker. And of course, body composition metrics. These are all really awesome indicators that are shining windows into biological age and giving insights into different systems and teaching us about how we're aging. But none of them tell the full story on their own. They're all incomplete. And this is also true for a newer category of biomarkers known as aging clocks, which I'm guessing people here are pretty familiar with. I've heard a lot of talk. I see a strong thumbs up there. Fantastic. So an aging clock is a computational model that predicts chronological age given molecular expression information. There are many kinds of aging clocks. A proteomic aging clock predicts chronological age given proteins. A epigenetic aging clock uses DNA methylation, and so forth. You can train these using machine learning, using artificial intelligence, using really simple computational approaches. But the key differentiator is you're taking molecular data and combining it in some way to predict chronological age. When you do a good job designing an aging clock and you apply it to a cohort, you see that people that are healthier are predicted to be younger. For example, people that exercise, that don't eat ultra-processed food, that don't smoke, are more likely to be younger than people that only eat ultra-processed food and are sedentary and smoke. And an especially well-designed clock is associated with age-related outcomes, such as future risk of chronic disease and how long someone lives. One thing that's interesting is that many aging clocks are not aging biomarkers because they're not associated with outcomes. Only a subset of aging clocks are aging biomarkers because they're providing more information than chronological age when it comes to teaching us about our own aging. So I've described a major challenge, which is that aging is too multidimensional and heterogeneous to capture with just one metric or biomarker. There's a separate challenge, which is really annoying, which is biomarkers often contradict each other across layers and within the same layer. And to illustrate this, let's look at an 81-year-old man who has publicly available DNA methylation data from whole blood. And what I've done is I've used the programming language R to predict this individual's epigenetic age using 7 different epigenetic clocks, all of which are suitable for application in adult whole blood. This table here is simplified from a paper under review and has 2 columns. On the left, the epigenetic clock used. On the right, the individual's epigenetic age. Rows in blue are where this person was predicted to be younger. Rows in red are where this person was predicted to be older. And what you see is this 81-year-old man is either epigenetically 43, pretty awesome, he's basically immortal, or he's epigenetically 89, not doing so great. This is a 46-year disparity. How do we make sense of this? Obviously, he can't be epigenetically 43 and 89 at the same time. And there's several takeaways from this, one of which the field currently is not in a good place when it comes to understanding how to take contradictory information and somehow weight it and put it all together. But also that even in the same layer of data, different biomarkers provide differential insights. And that also a clock should be specifically chosen for a very specific and relevant application. All right, so we've talked about these two challenges of aging being really complex, multidimensional. We've talked about biomarkers contradicting each other. But there's still plenty of room for optimism and excitement, despite this. We're now in a position where we've identified a huge number of metrics that provide useful insights into different systems. So despite there being a lot of unanswered questions, we're in a better position now than ever before to understand our aging. And this is why when you also survey longevity scientists, most of them feel that we can do a good job quantifying aging well, even if we don't understand everything about aging itself. There's a lot of optimism that this is a solvable problem. I share this optimism, and one of the reasons I'm very excited for the next few years is the amount of molecular data has been growing exponentially for many years now. And the quality of that data is getting better and better. If we take a focus on DNA methylation, in 2008, you could buy a methylation microarray for hundreds of dollars and get about 27,000 methylation sites measured. This is a really, really small amount of information because our methylome, which has all these chemical tags on it to regulate gene expression, actually has 28 million methylation sites. If you fast forward to today, for about the same price, you can measure about 1 million methylation sites. There's now a commercial methylation sequencing panel that captures 4 million sites, and whole methylome sequencing, all 28 million sites, is getting cheaper and cheaper. And this type of expansion is happening not just for methylation, but for the proteome and other omics layers. So one open question is, if we have access to the full methylome or proteome, just how good can we get at predicting future risk of death? It's not going to be trivial. Speaking of death, we're also getting better and better at finding ways to predict when someone is going to die in longitudinal data, adults tracked over time. If you go to the clock space, the first models that were released in 2013 and shortly afterward were either not associated with future risk of death or pretty mildly associated. In 2018, that changed when Dr. Morgan Levine and her colleagues came out with something called DNA-M PhenoAge, the first next-generation epigenetic clock that shattered all records for associating with lifespan and healthspan. Today, that clock is old news. We have many clocks that are more than twice as associated with future risk of death. So not only is the quantity of data increasing and the quality of data getting better, but researchers are getting more and more clever in their ability to build useful predictive algorithms. Ultimately, something I think about a lot is when I go to my doctor or someone goes to a longevity medical provider, that provider is somehow taking all of the information about you, your metrics, your conversations, your history, and compressing that into an idea of how you're aging. The doctor is basically a fancy AI model that's integrating many, many different layers of data. This is something that I think we can ultimately solve at scale with artificial intelligence using multimodal, multi-layered data. And it's going to be key that the model is actionable, it's responsive to change, that it's explainable, meaning that we can tell people why they're aging the way they are, and that it's also reproducible. You get the same result over and over. Dr. Max Shokurev, who's here with me, and I, as well as many other people in the space, are actively researching ways to make these models more powerful and explainable, and there's a lot of exciting work going on. And with that, I see I'm running out of time, so thank you so much. I appreciate you listening. Happy to take questions.
Yeah, thank you so much, Dave. That was really, really great. I think we have time for one question if someone wants to sprint to the microphone. I don't see anyone doing that. All right, yeah, thanks so much.
Thank you, Aubrey. Appreciate it.